Okay, so looking at RNA and the genetic code, a typical tRNA molecule has a cloverleaf shape when hydrogen bonds form between the complementary bases within the tRNA. The acceptor stem attaches to the amino acid and its anticodon bonds with a codon on the messenger RNA. So when I teach DNA, I teach it in steps. Coding is step 1, template is step 2, messenger RNA is step 3, and tRNA is step 4. Between each step, it's complementary and antiparallel. Okay, so that is right here too. So between the tRNA and the messenger RNA, it's complementary and antiparallel. RNA makes up most of the nucleic acids that are found in the cell, and it transmits the genetic information that's needed for cell operation. The molecules are polymers of nucleotides and differ from DNA molecules in 4 different ways. One is the sugar on the RNA is a ribose rather than the deoxyribose that's found in DNA. Then the uracil replaces the thymine because, like I said, it's a little bit of a diva. And then the RNA molecules are single-stranded, where DNA is double-stranded. And the RNA molecules are much smaller than the DNA molecules. So there are a couple different types of RNA. And the RNA is going to transmit information from the DNA in order to make the proteins, and it has different types. It has messenger RNA, step 3. And this carries the genetic information from DNA in the nucleus to the ribosome in the cytoplasm. Then we have tRNA, or transfer RNA, and this interprets the genetic information in messenger RNA and brings specific amino acids to the ribosome for the protein synthesis. Then we have ribosomal RNA, or rRNA, and this is the most abundant type of RNA, and it's combined with proteins to form ribosomes. So here is your study table, looking at the different percentages of the different types of RNA and the details between it. So you can see ribosomal RNA, or rRNA, is about 80% of total RNA, where messenger RNA is only 5% and tRNA is 15%. Okay? So looking at tRNA a little closer, the tRNA structures are similar and they consist of 70 to 90 nucleotides. They have a 3' end with the nucleotide sequence ACC, and this is known as an acceptor at stem. And they also contain an anticodon, and this is a series of 3 bases that complements the 3 bases on the messenger RNA. And how do we make proteins? Well, protein synthesis involves transcription and translation. So transcription in the nucleus occurs in the nucleus, and this is where the genetic information for the synthesis or making of a protein is copied from a gene in the DNA, and it's going to make RNA. Then translation is when the tRNA molecules convert the information in messenger RNA into amino acids, and these are then placed in the proper sequence to make a protein. So the genetic information in DNA is replicated in cell division, and it's used to produce messenger RNAs that code for the amino acids that are needed for protein synthesis. So looking closer at transcription. Transcription begins when the section of DNA that contains the gene unwinds, and an RNA polymerase enzyme uses one of the strands as a template to synthesize or make messenger RNA. mRNA is then synthesized or made using complementary base pairings, with uracil pairing with adenine in DNA, because remember, our RNA is a diva, so there are no T's. We instead are going to pair with a U. And then the newly formed messenger RNA moves out of the nucleus and into the ribosome in the cytoplasm and to be made into a protein. So here you have the sequence of bases on a DNA template. You're going to undergo transcription, and it's going to have the complementary base pairs in messenger RNA. Okay. Then during transcription, the RNA polymerase moves along the DNA template to synthesize or make the corresponding messenger RNA. And then the messenger RNA is released at the termination point. So you see that we have our DNA coding strand and we have our DNA template strand. And then when it separates, this is called the transcription bubble. Okay, and you see that we're using the template strand to copy off of and make that complement. So that's why the A will bind with the U, the C with the G, right? Is because we're working off of that step 2 template strand to go into step 3 messenger RNA. So between step 2 template and step 3 messenger RNA, they are complementary and antiparallel. So let's practice. What is the sequence of bases in messenger RNA that's produced from a section of the template strand DNA that has the sequence of bases etc. Okay, so pause me, think about it, and then come back. Okay, so remember, we're template strand is step 2, messenger RNA is step 3. In between each step, it's complementary and antiparallel, so we are going to match everything that we see. So that C will match with a G, the T with an A with a U because we're going to messenger RNA, A to a U, G to a C, and G to a C. All right, great job, you guys.